For the first part of our video, we will be discussing the phases and process of mitosis.
What happened?
The first step of mitosis is prophase. During prophase, we'll see the formation of the spindle apparatus at opposite ends of the cell, and the chromosomes will begin to condense within the nuclear envelope. Also during this process, the nuclear envelope will begin to dissolve. In the second step, prometaphase, the nuclear envelope is completely dissolved and the chromosomes are free to float about the cell. However, they get attached to the spindle apparatus by the microtubules. During metaphase, the next step, we see the chromosomes align in the middle of the cell. This structure is called the metaphase plate. If we take a closer look at each of the chromosomes, it looks like this, composed of two sister chromatids which are identical. Connecting the chromatids is a cohesion protein represented by the red bead, and the green beads are kinetochores which attach to the microtubules. During anaphase, each of the chromosomes will be pulled to an opposite end of the cell by the microtubules, and each chromosome is now considered a full chromosome. Moving on to the next phase, telophase, we'll see a formation of nuclear envelopes around each set of chromosomes, and during this phase, organelles will begin to replicate and the spindle apparatus dissolves. After telophase, the cell is ready for cytokinesis, where a cleavage furrow will form to begin the cell separation into two different cells. For the second part of our video, we will be discussing the process of meiosis. The first phase of meiosis is similar to the first phase of mitosis, which is prophase 1, where the chromosomes will condense, the nuclear envelope will begin to dissolve, and spindle apparatuses will form at opposite ends of the cell. Prometaphase is the second stage of meiosis, where the chromosomes are free throughout the cell because the nuclear envelope is completely dissolved, and microtubules will begin to attach to the chromosomes in the spindle apparatus. During metaphase 1, we will see the random assembly of chromosomes in the middle of the cell called the metaphase plate. These chromosomes are attached to the spindle apparatus by the microtubules. Take note that the chromosomes in meiosis are different than the chromosomes in mitosis. These chromosomes allow for genetic recombination, which is the exchange of pieces of chromosomes between non-sister chromatids, and the place where these chromosomes will cross over is called the chiasmata. Moving on to the next step, anaphase 1, we see that now the homologous chromosomes have been pulled apart from one another and randomly get pulled to opposite ends of the cell by the microtubules. Next, in telophase, a nuclear envelope will surround the chromosomes and the cell will have organelles replicated and prepare itself to be split. In cytokinesis, the cells will completely become two different cells with genetically unique results. To complete meiosis, the chromosomes will undergo the same steps of prophase, metaphase, anaphase, and telophase a second time. The results of this will be 4 sets of daughter chromosomes with genetically unique characteristics. And the cells will look like this. You have now learned the different phases and stages of mitosis and meiosis. Thanks for watching, and may the force be with you.